Hi, everyone. Thanks for joining in. My name is Kelly Marsh, and I'm going to talk to you today. Just let me show you my slides. And the fall, I hope I can see everything. You can see everything here. But I'm going to talk to you today about PeakML. But before I dive into the development of that, I just wanted to give a little bit of background about my company. So I work at Agios, and at Agios, we are focusing on developing precision-based medicines for cancer and patients with rare genetic diseases. We have over 550 employees, 2 approved molecules, and we have several programs that are in the clinic, and we're excited about those. Our expertise and discovery platform is really centered on and focused on 3 fields, those of cancer metabolism, metabolic immuno-oncology, and rare genetic diseases. I'm in the Metabolomics and Proteomics group at Agios, and we are comprised of a diverse skill set of individuals. So we have mass informaticians, bioanalytical chemists, as well as those that are formally trained in biology and informatics. And that really allows us to execute our goal, which is the design and execution and analysis of all of our in-house omics data, including metabolomics, lipidomics, and proteomics. We spend a lot of time and energy, as you can imagine, maintaining and building our platform. This is the front end, the sample preparation, as well as the mass spec instrumentation, and including the back end, which is the data processing and analysis pipelines. We're integrated across a research organization and work with drug programs all the way spanning from the early stage and the target identification or discovery mode all the way through the preclinical drug phases. And as such, and because of this, this does require for us to have capabilities to conduct a wide range of experiment workflows that when we need to measure lipids and metabolites. For example, sometimes we just need to look at a specific chemical reaction in the cell, and this is just honing in on those enzyme precursors and products. We do a lot of work also when we're looking at a pathway flux analysis, and this is when we utilize stable isotope tracers to measure all of the intermediates within a pathway. We also conduct experiments where this is more broad profiling or more of that omics space where we're trying to capture as much of that chemical space as possible. And this is when we're looking and conducting experiments for biomarkers or really characterizing a new model system. And over the past 9 years that I have been at Agios, one major shift since those early days has been within this data acquisition step. And this is in principle a result of the advancements in mass spec-based technology with the commercialization of more of these high-resolution accurate mass instrumentation, specifically the Q-Exactive with the Orbitrack technology from Thermo. Adoption of these instruments into our pipeline has really changed the way that we acquire data. And the scan speeds, the resolution, the mass accuracy has improved to a point that there's less of a trade-off between precision and accuracy. And sorry, I just saw that I'm running along. Less of a trade-off with precision and accuracy, and you're not worrying about sensitivity as much. So with that, we really now utilize sort of these workhorse methods that, while not for all of the samples, but for most of the samples that are analyzed in our lab, we use this standard method, and we operate the mass spec in this full scan mode. So what that is we tell the mass spec to capture all of the information across the mass range. For us, in metabolomics, that's anywhere between 7 to 700. And what this effectively means is that it's really just the data processing and analysis step that's tailored to the type of experiment workflow that you're on. So even when you're only looking and doing these targeted analysis and only pulling out a handful of metabolites, you're still extracting that information from these large comprehensive data sets where obviously most of it is just not being looked at. And that is mostly because regardless of the software they're using, whether it's LMaven, which is what we use, or other commercially or publicly available untargeted dataset platforms. The activation energy to engage in this is quite high, and that's because this workflow is much more time consuming, not as straightforward, and more labor intensive. So we talked to Elucidata about this problem, and they shared our passion for really developing a better pipeline for this untargeted workflow, and also trying to better create a data analysis software that was a better match for this advancement in technology and our ability to acquire the types of the data that we're acquiring now. So about a year ago now, we set out on a collaboration with the Elucidata team to answer the question, can we develop a semi-automated workflow for non-targeted metabolomics analysis? And this was a clear application for machine learning to help us do just that, and not just for future untargeted analyses, but perhaps even for retrospective analyses for data that we just have sitting on our server. So I'm going to talk to you a little bit about how we did that, and that will involve me sharing with you what I think is the unglamorous side of mass spec data analysis, at least in my opinion, and that deals with peak curation. So you run your analysis over your chromatography on capturing all that mass information across the, as it elutes off the column. So then you have to sort of interact with the software in a stepwise manner and identify all of the peaks that you see across all of the mass range, across all of that time dimension. And you'll often see peaks that look like this, and you have to tell the software that indeed this is a real signal and you mark it as good. And you have to distinguish that from peaks that perhaps look like this, and you know, this is noise and please ignore that, and you mark it as bad. It turns out that there's also this huge gray area where you see peaks that look something like this, and this requires sort of a little bit more expertise, or even just a gut call here based off of the instrumentation that you're working with to decide, you know, which category that falls in. So the Elucidata team wanted to develop this, and they're calling this PeakML. And ultimately, what this software is going to do is the peak curation step and classify the features as either good or bad. And so how we did that was that we needed to come up with a way for the machine learning algorithm to classify the features of a peak. So the Elucidata team came up with 88 different peak attributes that are mathematically describe sort of basic features of what a peak is. And these include Gaussian shape, signal to noise ratio, symmetry, as well as even asking the question, you know, is this peak found in any other similar cohorts across your sample? The next step was choosing an algorithm. And for that, there's a lot of machine learning algorithms out there, each with their own pros and cons. And ultimately, we landed with XGBoost. There were several reasons for that, but one of which was the model interpretability. We wanted to be able to see the decision tree and the attributes that were driving the classification that was that the machine learning algorithm was doing. We felt that was important. So with this framework and the algorithm, we then just needed to train it and to give it data sets so that it could learn what a good peak looks like and what a bad peak looks like. And for that, we collected around 6,000 peak groups and features across, 16 different data sets, multiple different types of chromatography, different back-end mass spec instrumentation, and we went through the training and validation, which is just sort of this iterative stepwise process. And I'm just going to walk you through a couple things that we learned about this along the way. So the first had to deal with user-to-user availability. So myself and were colleagues that I could recruit for this project, I asked to curate here 5,000 different peaks. And we said, okay, we're going to take the data set where we all agree, and we're going to use that to train the model. Well, when we looked at the data after everyone completed it, we were a bit surprised to see that only approximately 50% of the time, sometimes a little less, sometimes a little more, were we agreeing on whether a peak was good or bad. And that was a bit surprising. So when we looked at the data that was giving rise to this, and we did a dimensionality reduction plot to really understand that. You can see that the real signal in blue and the orange signal in noise really clustered into these distinct populations. This is suggesting that these are the peak attributes that are driving whether this is a good or a bad. Everyone is agreeing on this, and they're clearly distinct populations, and that this is what I would refer to as obviously good peaks and obviously bad peaks. So it makes sense that we're all agreeing in agreement there. Instead, when we looked at sort of this fraction of peak where there was more disagreement, so 4 of us felt one way and 2 of us thought it was the opposite way, you can see that any sort of clustering falls apart. So clearly these are what we refer to as those like ambiguous peaks. So it makes sense that we weren't all in agreement there. And it was really important, and we've been spending a lot of time to kind of go back and generate consensus around these peaks, these ambiguous peaks, because if we just use the data where everyone agreed on, we would be at risk for developing a machine learning algorithm that was just really good at identifying obviously good and obviously bad peaks. And that's not what we want. We want to give it a sufficient training data set that can really make those harder decisions about those ambiguous peaks. And then one could imagine that, you know, as we get new scientists, as our group changes involved, as we get new and different instrumentation and technologies, making those decisions about what is a good peak and what is a bad peak can change. So that brings me to my next thing that I wanted to share with you, which is learning about how this the algorithm can learn new user behavior. And to do this, we set up and compared 2 different frameworks. The first is on the bottom here, is essentially when you're starting from scratch. So you have the framework and the algorithm, and you just give it a bunch of, you know, the new user, it gives it a bunch of curated peaks, and you train the model that way. And that's referred to as a personalized model. The second, and in contrast, is where we take that global model that was built at Elucidata, and we use that as the base model. And then we just ask the user to input, you know, a very small, and we determine this to be around 200 peaks of a balanced data set. And this allowed for the model to be sort of a continuous learning one. And when we compared the accuracy from the continuous learning to the personalized model, what we found is that they were quite comparable. In fact, if anything, the continuous learning model actually outperformed the personalized model. And this was important because it suggested that the framework was able to sufficiently capture that new user behavior, but benefited from that retaining the information from the global properties, and ultimately only required that new user minimal effort for it to be trained. So for this, it's you know, we're excited that this is scalable and adaptable. So now that we have the model and it's trained, the next thing was to validate it. And to do that, we did sort of this human versus machine head-to-head comparison. And the expert chosen, which was loosely defined, was me. And I went up against PeakML, and we wanted to see how we performed. So the same data set was loaded into LMaven. The peaks, the features were detected, and then I went manually and went through approximately 3,500 peaks to choose whether this was good or bad. And we compared that to what PeakML did, and the results are here. So the first thing we looked is at accuracy. So assuming everything that I did was valid and true, we then asked how often did PeakML match my answer for those peaks. And of course, there were some false negatives and false positives, not surprising, but overall, the accuracy was around 94%, which we were quite pleased with. In addition, it was much faster than me. So for around 3,500 peaks, this took about 10 hours of my life. And this was not consecutive because I can never sit down for that long in a row and do this. So this spanned over 4 to 5 working days. And so this is an entire work week that would take me to kind of turn around this type of data. In contrast, the machine learning algorithm could do this entire step in less than 4 minutes. And we think that's important, both for, you know, obviously it takes less time, but this allows the user to sort of do this iteratively if you're not happy with the parameters that it used. Finally, it detected around 2.5 more features. So this is important. So, you know, in order for a scientist to engage with these data sets, you need to have a manageable data set where you defined your baseline and define what the minimum signal you're going to look at is. obviously those limitations don't apply to the computer, so it can really have use less conservative thresholds, and doing so, it was able to identify an additional 5,000 features. So this is another 5,000 ions, which are potentially 5,000 other metabolites or molecules of interest that could have significance in my experiment, much more low-level signals. So we were excited about that. And it doesn't stop there. So the PeakML actually does what I kind of refer to as like the second tier type of analysis. So in addition to the binary good or bad, it will then ask questions and try to identify additional information, including covariance. So does this peak that it found, does the signal intensity vary across all of your samples? Because if it's varying, that means it's changing, and that could have really important impact upon the drug treatment or whatever you're looking at. And it'll flag that as a blue star. Also, if it's correlating with any other peaks in the sample, it'll flag that as a yellow star, and that's important as a metabolomic scientist. That can tell me information about whether this is an adduct or a fragment. Obviously, you get this hybrid model if it's both of those, and if it's none of those and it's just a regular good peak, you'll get the green check mark. And I just wanted to share, this is a snapshot of what the user interface looks like. And as you can see here, this all happens within the LMaven software. So we now have a column for the model prediction probability. Here are all the icons that I just explained to you, the correlation widget and the covariance widget, and where you can filter out the data to identify any of those that you want to take and manually interact with. And so we're really excited about, where we are. We still have some things to work on and fine tune, but we're really pleased that sort of this product, this project and where we are, and where we designed, developed, and validated a machine learning algorithm that was fast and quite accurate, and it's all integrated with the LMaven platform. And we really tried to make sure that the user interface was such that the scientists could get from raw data to biological insight within minutes. So with that, I just wanted to thank the team at Agios, and this is the only way that I get to see them these days, but thank them. This is an amazing group of people that I'm lucky to work with, and especially want to thank Tom, who's my boss, for the opportunity to work on this project. And of course, I need to thank the amazing team at Elucidata that has just been incredible to work with on this project. Saiful and Sakshi did a lot of work integrating PeakML into the LMaven platform. Kailash and Shashank did all of the model development and validation, and Richa for doing everything, keeping us on our toes. And actually, if it wasn't for her, this project would not be where it is. So a huge thank you to her. And I'm sure that Abhishek did something at some point. So just wanted to thank him. But with that, I just wanted to thank you for the opportunity to speak with you today. And I'm happy to take any questions. Did you look at the mass of the ion and whether there were the expected isotopes and adducts? It's not just whether a peak looks real or not. I think I understand what this question is, so I'll take a shot at it. So you're absolutely right in that PeakML is just sort of on that first step of whether it is a peak or not. As a metabolomic scientist, I only really want to look at metabolites. I don't want to look at plastics. I don't want to look at contaminants. I don't want to look at other things that are just, in my sample that, of course, contribute to peak. So anything that would need to look at the spectra or look at adducts or annotation for that is going to happen downstream. And I'm actually working with Elucidata on those next steps. But right now, the PeakML is just that first initial step of weeding out true electronic noise rather than contaminants and not and artifacts rather than just biological artifacts. I hope that's what you were asking this. Thanks for the tip about the analytical chemistry publications. I do think we looked at these, but I will make sure I go back and make sure that those were some of the articles that we reviewed. How long did it take to train? I answered those. It was 10 hours of my life, and I was kicking and screaming the entire time. How many? Okay. How many data sets were used for the base model? So at this point, it's around 6,000 features. The Agios model that we're training is around 5,000, but we're going to give it more. I think I got through all of the questions. So if there are no more, then I can end the session because we are definitely over time. Thanks, everyone. And so how we did that was that we needed to come up with a